Hello class and welcome back to part 203 for Unit 6 exam overview. So here we're moving on. Video 1 is question 1 through 10 with a little break hopefully right after. And right now we'll be working on the next 10 questions. So I definitely understand this was one of our longer exam, and that is the push to keep working like consistently and quickly. A lot of us, what I noticed during the exam is we were putting our head down, or we waited till the very end to try to finish the exam, and that is going to affect us. If we do that on the start, we're going to be stuck in the same situation. So again, make sure that you're working quickly, you're working promptly, so you can get that information completed. So again, moving on with question 11, which of these is a direct result of an error in the transcription of DNA nucleotide? So remember, transcription is when DNA goes to messenger RNA. So again, here's if we have the message wrong. And let's get started. So again, the nuclear membrane is ruptured. The nuclear membrane surrounds the nucleus, right? Nuclear membrane, and rupture means to break. Having the wrong information does not do anything to the membrane, so that is out. The amino acids do not bond to tRNA. Here, that does not have anything to do with the bonding of amino acids to tRNA. That already comes made, so we already know that is also not our answer. The codon sequence is incorrect. So if we have an error, does that mean our codons are wrong? That could be correct. Remember, DNA, everything is in codons or in 3 letters, and the transportation of mRNA does not occur. So mRNA is going to take the message no matter what. Nothing is stopping that transportation that we know of, but it does give you the wrong message. So our message is in codons. The codon sequence is incorrect. Yeah. So if there is an error, do we send the wrong message in the codons? Absolutely. This is more vocabulary based, and critical thinking, guys. Just process of elimination. Take it slow, and make sure you're reading the questions and answer choices carefully. Remember, I know you're taking your exam online. You do have that cross it out option. Which of the following is not a function? That means it is not a job of nucleic acids during transcription and translation process. So which do our nucleic acids not do? Not a job. Carry genetic information from the nucleus to the ribosome. Yeah, this is transcription. That is definitely a job. Serves as the source of information that remains secure in the nucleus. So yeah, nucleic acids, which makes up DNA, do have that information. The big clue is remains in the nucleus. Nucleic acids, DNA, carries amino acids that are bonded together to form proteins. This is translation. So again, carrying amino acids to make proteins is specifically tRNA or transfer RNA. That whole process is translation and forms the nuclear envelope to protect genetic material. So again, the job of nucleic acids during transcription and translation, this is protein synthesis, making proteins, right? Forming a nuclear envelope to protect genetic material, that is nothing. That is our false statement, right? Remember, we're looking for something that is not true. That is not a job of nucleic acids. DNA does not create its own nuclear envelope, especially not during protein synthesis, which is transcription and translation. So again, this is just process of elimination and taking your time. I can see how both of these could be a little bit more confusing, especially if we struggle with the language. But again, process of elimination, use that to help you. So, the easy one. A student builds a model of a DNA strand. We know we have DNA because that thymine is there. Which of these models of DNA shows bases that are complementary to the ones in the DNA model? So we're going from DNA to DNA. We want our DNA complementary strand. So remember, at the golf course. So let's split it up into codons. We have CTA, CCG. We want to make the complementary strand. So C binds with G, T with A with T. Then we have GGC. We're looking for this strand on our answer choices, the complementary DNA strand. So we have GAT, CAG, A is out. GAA, nope, we're looking for GAT, B is out. GAT, yep. And then we had, let's go back, GGC. Yes, both of these are correct. So this is our DNA complementary strand. As I mentioned before, this is a question that they like to ask. It's super easy, papitas. We already know at the golf course or adenine and thymine, guanine and cytosine, those are our base pairs. So again, they give us two codons, CTA, CCG, and we just find the complementary strand, GAT, GGC. So this one should have been easy, and our answer was C. Ooh, this next one, we already see a quick image here. We know that looks like a T, so that's a big clue. And these are amino acids. Again, be very observant, and we see mRNA right there. So a process is shown, a biological process is shown. What is the purpose of this process? So why is this process occurring? We see mRNA and we see amino acids. So we should be getting a real big clue, mRNA to amino acids, that this is translation. We're looking at an image of translation. This is your tRNA, and it actually does look like a T, so that is a big giveaway. This is a tRNA with the anticodon binding to the codon on mRNA and making our protein. So to replicate DNA, I do not see any DNA anywhere. This is messenger RNA, and that is not DNA at all, so A is out. To assemble nucleotides in mRNA chain along a DNA template, again, they mention DNA. DNA is not here. So A and B are automatically out. To synthesize, to make amino acids used to unzip strands of DNA and copy the genetic code. Again, they keep mentioning DNA. We are not unzipping any DNA at all. To translate, we did say that was translation, the genetic code into specific sequence of amino acids. If we go back, you see our tRNA and we see our amino acids here. We know they're amino acids because they're that 3-letter combination. Absolutely. D is what is happening in that image, the translation process. So we have the mRNA, the genetic code is specifically messenger RNA, and we're transcribing it to a specific order of amino acids. So our best answer was definitely D. That is what you see on these images. The messenger RNA has the codons or the instructions, and what the tRNA does is that it attaches itself and it creates the amino acid chain. The amino acid chain makes the proteins, and the proteins make the traits of how we look. So again, it's just like playing telephone. DNA sends the information to mRNA. mRNA sends it to tRNA to make the protein, and that's how we look. Question 15. Which of the following mutations would have the potential to affect future generations of a species? So future generations is the offspring. It's something that you are passing down to your babies. So how can a mutation affect your offspring or your babies? Remember, your offspring is going to be made of an egg and sperm. That is the only thing that you give your child, the only type of cell. Everything else, your cheek cells, your eye cells, anything like that, that is called somatic. So somatic cells our body cells. Those are your own. And then you have gametic cells or gametes, and those are the ones for reproduction. So it's either an egg or a sperm, and that is the only thing you pass down to your child. So let's start looking at process of elimination. So which of the following mutations would have the potential to affect future generations? A frame-shift mutation on the X chromosome of a cheek cell. So again, the cheek cell is our cheek cell. The cells that we have in our cheek are not going to get passed down to our child, so that one's already a no. A chromosomal mutation on the Y chromosome of a kidney cell. Again, your kidney, your problem, essentially, that's not getting passed down anywhere. A point mutation on the first chromosome of a sperm cell. A sperm is something that you do give to your child, right? That is a gametic cell. So that is definitely a possibility. And a substitution mutation on the third chromosome of a uterus cell. So your uterus is this organ where a baby grows in, right? You do not give your uterus cells to your child. You're not going to give it to your son, like something like that. So using process of elimination and what we know, that the only way to pass on a mutation is through your gametic cells, through your egg or your sperm, your best answer is C. So the only way to pass down a mutation again to your child is either through the egg cell of a female or the sperm cell of a male. Process of elimination, super easy. Question 16, what is the function of the nucleic acid in messenger RNA? So what is the job of messenger RNA, right? So it carries genetic information from the DNA to the ribosome. I mean, yeah, the messenger RNA is a messenger. It's taking a message from DNA to ribosome. It carries amino acids. So mRNA does not carry amino acids to the ribosomes, not at all. This is actually tRNA. It facilitates chemical reactions that form ribosomes. We do not form ribosomes. That is actually rRNA or ribosomal RNA. And it serves as the site of protein synthesis? Absolutely not. Remember, protein synthesis takes place basically in the ribosomes and a little bit in the nucleus when we get the information. So by process of elimination, and honestly, just knowing your vocabulary, this was in the review. What is the job of mRNA? It is to carry the genetic information from the DNA to the ribosomes. It's taking the message. So that is our best answer. Nitrogen bases are located on both strands of DNA double helix. What is the significance of the nitrogen bases? So the number of adenines and cytosines determines the type of RNA that will be produced. We have not heard that at all. The order of nitrogen bases determines the order of amino acids in protein synthesis. Well, yeah, the way that the bases are in order, because remember that gives us our codons, that makes our amino acids, and that determines what proteins they are. The amount of thymine and guanine in DNA molecules determine the length of the genes. Not really. And what you should notice about these answer choices is that you have A and C, which are not matching base pairs, and you have T and G, which are also not matching base pairs. So again, the nitrogen bases located in DNA determine the type of hydrogen bonding between the bases, which amino acids will be added to the polypeptide chain. That is also incorrect. So there is only one type of hydrogen bond, a plain old hydrogen bond. So that is what makes it incorrect. So yes, the nitrogen bases located in the DNA. So the order of these bases determine the amino acids and the traits. It goes back to, I think it was one of the first couple of questions. The order of your nucleotides determines your traits. That order is going to make your amino acids and it's going to make your proteins. Very important key point to understand. Ooh, here we have another process occurring. So during DNA replication, A chemical called DNA polymerase acts as an enzyme to quickly bond nucleotides to the complementary strand. DNA polymerase belongs to a large class of biomolecules that perform certain actions, right? So this organism is called. So we already know this. We've studied this a lot. Enzymes are what biomolecule? Are they carbs, lipids, proteins, or nucleic acids? Remember, enzyme is a special protein that speeds up or catalyzes reactions. So our best answer for this one, class, was definitely proteins. So during DNA replication, where you're making a copy of DNA, you see it right here, the double strands. Again, you're using an enzyme, and an enzyme is a protein. Ooh, okay. So here it says we have an mRNA sequence, and we can tell because we have that uracil there for us. We have our chart here. Let's look at the question really quickly. Oh, okay. So we're looking at the first. Your teacher forgot how to count. You're looking at your first 4 amino acids. So let's go back and let's get started. So we have AUG. So AUG. We have methionine as our first one. Remember, it was just our first 4 codons. We have methionine. GUU. Here we have GUU. Valine is our second one. Triple A, so we have A, and A, that is lysine. Again, it's just using your chart, your first base, your second base, and your third base. And the 4th codon was CGA, so here is C, here is G, and here is A, arginine. So methionine, valine, lysine, and arginine. Methionine, oh, I already forgot it. Valine, lysine, and arginine. You'll just have to correctly choose the amino acids by using your chart, which is something we've practiced before. Again, it tells you here that it already gave you the mRNA sequence, and the other clue is the U. So you did not have to make a new strand. If you made a new strand, then that's probably how we got the question wrong. Here we go, another mutation question. So sickle cell shaped red blood cells result from a mutation in the gene that codes for hemoglobin. So we know it is a change, a mutation. The mutation results in sickle cell anemia. What type of mutation is depicted in the sequence? So I know I had people ask me like, what is happening, or what is sickle cell? We don't really need to know the specific of sickle cell to answer this question, but sickle cell is when the shape of the red blood cell is changed and it cannot carry a lot of oxygen. So changing the shape of your red blood cell reduces the amount of oxygen being carried, and it can cause a lot of health issues. So again, going back to the mutation, on our original or our normal hemoglobin, we have 1, 2, 3, 4 codons. On our sickle cell, remember, codons are 3 letters. We also have 4 codons. So insertion means to add, deletion means to subtract. These are 2 I can automatically cross out. I'm not missing any base letters, and I do not have any base letters that are extra. So I'm good on that end. T, G, and A, our first codon is exactly the same. G, G, T, again, exactly the same. C, here we have a difference. TNA, CC, and then CTC. So codons 1, 2, and 4 are exactly the same. The only change happens on codon 3, base letter 2. Originally, we have a thymine, and in the mutation, we have an adenine. So again, all they did was change the letter, so that is a substitution. That is the type of mutation we see there, and that is a type of mutation that causes sickle cell. We know it is not a frame shift because only that one codon changed. The ones before and the ones after stayed exactly the same. Remember, frame shift, everything changes. Everything around it changes. So again, hopefully all of this information is helping us understand what we should expect to see on Unit 6. I am going to end this video. The last part will be right up. It'll be the last couple of questions. For this break, it'll be a little bit shorter, maybe 2 to 3 minutes in case you need to go to the restroom or get a quick water break. But thank you so much for coming to Saturday school, or if you're reviewing this video at home because you couldn't attend, thank you so much, class. Please make sure that we are studying. Again, all semester review material is in Teams. It was sent out on Remind, so all the information will be there. Best of luck studying, and I'll see you for the last part of this video.